Yes, Assalamu alaikum, dear students. How are you? I think many people have joined the meeting. Let me start. Today we are going to start our first portion, and this portion is about cell biology. We will study here the different aspects of the cell, especially with reference to the objective point of view. No doubt you have studied the cell in the ninth class, 10th, FSC, and different terms or semesters of your VS program. But now study cell biology with relatively a different way, that we will study this with respect to their objective value. So here is some data which I have collected from different books. This data, as you can see here, that this data is related to the work of different scientists or contribution of different scientists regarding the cell or cytology. Let me start. Bismillahirrahmanirrahim. In 1665, Robert Hooke reported cell in his famous publication Micrographia. You have to remember here three things. Number one is the year 1665. Number two is the scientist who first of all observed cell was Robert Hooke. Number three, the name of the publication was Micrographia, and he observed cork cells under his microscope. This microscope was his self-made microscope consisting of two simple lenses which were placed one above the other in relatively flexible way so that they can be adjusted to focus the image. And he observed first of all cork cells, and as you know, the cork cells are dead cells. The first cell which were discovered, which were observed, were dead cells. So all these appeared in the form of honeycomb like structure, and it gives it gave a misconception regarding the cell, that the cell is an empty space with with nothing inside it. Then in 1805, Lawrence Ocken, a German biologist, said that all living beings originate from or consist of vesicles or cells. This was an important concept, that living beings originate from living things, because before this, the concept of abiogenesis existed, and abiogenesis the concept of abiogenesis explains that all living organisms may be produced from non-living things in specific conditions. जैसे कि लोगों को ये कंसेप्ट था कि किसी जगह पे अगर पानी खड़ा रहे तो वहाँ पे मेंढक खुद पैदा हो जाता है, किसी जगह पे अगर जो है गोबर वगैरह पड़ा रहे तो वहाँ बिच्छू खुद पैदा हो जाते हैं, गोश्त किसी जगह ओपन पड़ा रहे तो वहाँ पे सुंडियाँ खुद पैदा हो जाती हैं। तो ये कंसेप्ट क्या कहलाता है, ये बायोजेनेसिस। इसका कंट्रेरी कंसेप्ट जो है वो इनिशियल स्टेज पे लॉरेंस ओकन ने दिया। नो डाउट ये उसने कंसेप्ट दिया, लेकिन इसके बारे में जो जो बेहतर काम, या यूँ कह लें कि एक्सपेरिमेंटल प्रूव जो था, वो खैर लामार्क उसने सॉरी पास्चर ने जो है वो दिया था अपने बैक्टीरियल रिसर्च के ज़रिए से। Next, in 1809, Jean-Baptiste Lamarck said that no body can have life if its constituent part are not cellular tissue or are not formed by cellular tissue. There are two concepts. Number one, that life or living things consist of two things. Number one is that their bodies are composed of cellular tissues, and number two, some material may be produced or formed by cellular tissue, but it will not be a living tissue or living structure. For example, our nails and hairs, these are not living structures, but these are produced from cellular tissue. So the body of the organism will be composed of two different things, either cells or the things which are produced from the cells. Next, in 1831, Robert Brown discovered nucleus, and this was another important discovery which changed the concept of the cell as an empty space. And now the cell is considered filled with something, and this gave a way to the scientists that new research should be conducted to find out more different structures which are present inside the cell. And next, in 1838, Schleiden, a German botanist, and in 1839, Schwann formulated cell theory. Next, in 1855, Rodolphe Virchow said that all new cells only arise from previously existing cells, and in German language, omnis cellula is cellula. He said that all cells always arise from pre-existing cell. There is no प्रोडक्शन ऑफ स्पॉन्टेनियस प्रोडक्शन ऑफ सेल्स. ये स्पॉन्टेनियस जो हम वर्ड यूज़ कर लेते हैं, स्पॉन्टेनियस प्रोडक्शन मतलब तो खुद ब खुद कोई सेल वगैरह पैदा नहीं हो सकता. Then in 1857, Albert von Kolliker discovered mitochondria. This was the discovery of mitochondria, the powerhouse of the cell. Richard Altman in 1866 used the term bioblast for mitochondria. Then Carl Bendan, 12 years later, renamed it mitochondria. These are different concepts or discoveries of related to the mitochondria, uh, m- uh, which are here in the same sentence, irrespective of the years, which are arranged here in the upper and lower sentences. And next, in 1862, Louis Pasteur provided experimental proof of the biogenesis by working on bacteria. तो ये मैंने अभी बात आपको बताई कि बेसिकली इसको प्रूव किया था लुईस पास्चर ने. इसमें देखिए ना वर्ड जो बायोजेनेसिस है, बायो फॉर लिविंग ऑर्गेनिज्म, जेनेसिस जहाँ भी वर्ड आता है प्रोडक्शन के लिए आता है. लिविंग थिंग्स से लिविंग थिंग्स का बनना क्या कहलाएगा, इसको हम बायोजेनेसिस कहेंगे. नेक्स्ट इन 1863 Swiss biologist Rudolf von Kolliker used the term cytoplasm, and in 1880 August Weismann said that all cells have common origin. फिर इसके बाद देखेंगे इन 1898 की मिलोगॉल्जी डिस्कवर्ड गॉल्जी अपरेटस. इन 1925 गार्टर एंड ग्रैंडल proposed bilayered membrane model of plasma membrane. According to him, membrane consists of two layers of lipids only. There is no presence of protein in it. This was an initial concept which was changed later on. And next, in 1928, Otsolf reported the presence of cytoskeleton, and in 1977, Cohen confirmed the presence of work of Holtzow through electron microscope. This was a much later work. And next, in 1935, Hillary and Davison proposed three-layered model of plasma membrane. Yeah, and I am editing something here that this was the model regarding plasma membrane. Okay, next. In 1945, Keith R. Porter, Claude, and Thompson discovered endoplasmic reticulum (ER). 1955, George Plaidy discovered ribosomes. 1959, Robertson proposed unit membrane model, and in 1961, Beaver and Bradenspach discovered glyoxysomes. Next, in 1972, Singer and Nicholson proposed fluid mosaic model. Next, in 1974, Christian Didier discovered lysosomes. He won Nobel Prize in Medicine. This was the contribution of different scientists regarding the cell. And next, we will move towards the basic concepts of the cell. Sorry, I had a call. Sorry. Okay, let me move towards the next concept, difference between prokaryotic and eukaryotic cell. Uh, you have studied a number of differences between prokaryotic and eukaryotic cells, and next we will move further and we will take a in detail view of the differences between prokaryotic and eukaryotic cells, and especially with reference to the objective point of view. Number one, terminology of prokaryotic and eukaryotic cell was initially introduced by Hans Riss in 1960, and prokaryotic cells include. These are just different concepts which are here in the form of simple sentences. First of all, you have to remember the prokaryotic cells, bacteria, and bacteria include archibacteria, eubacteria, and mycoplasmas, all different groups. And number two is of cyanobacteria, second group. Second group, cyanobacteria, as we have discussed in the initial orientation lectures, that cyanobacteria were previously known as blue-green algae. These were placed in the algae, then they were replaced from that area because these have prokaryotic cells, and now they are placed in the kingdom prokaryotic because these have prokaryotic cells. So basically, there are two groups of prokaryotic cells, bacteria and cyanobacteria. Other than these two bacteria, a simple concept I am describing here, other than bacteria and cyanobacteria, all other cells are eukaryotic. Okay, like eukaryotic cells include cells of plants, animals, algae, fungi, etc. All these cells are eukaryotic. Only bacterial and cyanobacterial cells are prokaryotic. Okay. Next, the internal organization of eukaryotic cell is much more developed as compared to prokaryotic cells from which they are believed to have been evolved. There are two concepts in this sentence. Number one, the internal organization of eukaryotic cell is much more developed because of the presence of membrane bounded organelles. देखिए हम जो है ना उमन प्रोकैरियोटिक सेल यूकैरियोटिक सेल जो किस बिना पे कह देते हैं, अब ये मैं अहम कंसेप्ट आपको बताने लगा हूँ कि प्रो का मतलब हम कह देते हैं जी कि क्या है, बिफोर अर्लियर कैरियोटिक कैरियन है न्यूक्लियस, तो इसमें विदाउट न्यूक्लियस का कंसेप्ट है, बिफोर न्यूक्लियस का कंसेप्ट है। ये ओल्ड सेल्स हैं जो कि अर्लीस्ट सेल्स हैं जो कि बने थे हमारे ज़मीन पर जिस वक्त ज़िंदगी का आगाज़ हो रहा था उस वक्त। लेकिन ये है कि हम उनको इनको जो है ना मतलब कंसिडर करते हैं कि ये वेरी इनिशियल फेज़ के सेल्स थे। इसके बाद जो सेल्स बनना शुरू, उन्होंने न्यूक्लियस बना लिया। इस बिना पे अब उनका बाद वाले सेल्स का नाम क्या होगा, यूकैरियोटिक। यू फॉर ट्रू, कैरियोटिक कैरियन फॉर न्यूक्लियस, हैविंग टू न्यूक्लियस। अब इससे ना हमारा एक कंसेप्ट क्या बन जाता है कि व्हाट इज डिफरेंस बिटवीन प्रोकैरियोटिक एंड यूकैरियोटिक सेल। डिफरेंस लाइज इन द प्रेजेंस और एब्सेंस ऑफ द न्यूक्लियस। ओके, but remember, the difference between prokaryotic and eukaryotic cell doesn't lie only in the presence or absence of the nucleus. Rather, all membrane bounded organelles are absent in prokaryotic cell and these are present in eukaryotic cell, like other than nucleus, mitochondria, chloroplast, endoplasmic reticulum, Golgi complex, etc. All these are membrane-bounded organelles. All these are absent in prokaryotic cell and these are present in eukaryotic cell. Okay, so eukaryotic cells have variety of different organelles, like all membrane-bounded organelles, all non-membrane-bounded organelles are present in eukaryotic cell, while prokaryotic cell is relatively simple because it lacks all membrane-bounded organelles. Okay. Next, the other concept is that they are believed to have been evolved. Mean it is a general concept that prokaryotic cells are ancestors of eukaryotic cells. And next concept, all membrane-bounded organelles like nucleus, mitochondria, chloroplast, endoplasmic reticulum, lysosome, vacuoles are absent in prokaryotic cells, along with centrosomes and microtubules. Why I have highlighted it? This is an important concept. Centrosomes are centrioles. These are non-membrane bounded organelles. Centrioles are non-membrane bounded. They do not have membrane, but still they are absent in prokaryotic cell. होना तो ये चाहिए ना कि जो प्रोकैरियोटिक सेल है उसमें मेम्ब्रेन बाउंडेड ऑर्गेनेल्स ना हो, तो बाकी जो नॉन मेम्ब्रेन बाउंडेड हैं वो तो मौजूद हो ना। तो लेकिन यहाँ पे नॉन मेम्ब्रेन बाउंडेड ऑर्गेनेल में से अगर आपसे एमसीक्यू पूछ लिया जाए कि वो कौन सा नॉन मेम्ब्रेन बाउंडेड ऑर्गेनेल है जो नॉन मेम्ब्रेन बाउंडेड होने के बावजूद प्रोकैरियोटिक सेल में भी एब्सेंट है, तो आप क्या जवाब देंगे? सेंट्रियोल्स या सेंट्रोसोम, जो मतलब जो है ना, जो भी इसमें ऑप्शन दिया वो। माइक्रोट्यूब्यूल्स मतलब हो गया साइटोस्केलेटन, तो ये भी जो है इसमें सेल में प्रोकैरियोटिक सेल में एब्सेंट होता है। तो ये इंपॉर्टेंट कंसेप्ट है, आपने याद रखना है। ठीक है। Next, we will move towards the table. Table indicates the differences between prokaryotic and eukaryotic cell. First of all, a prokaryotic cell is surrounded by a single membrane layer, mean only a single membrane in the form of plasma membrane is present outside the cells, while in case of eukaryotic cell, some eukaryotic organelles are bounded by double membrane layer. For example, nucleus, mitochondria, chloroplast, all these are double membrane bounded organelles. Next, number two, in most cases, the cell wall surrounds the plasma membrane and is composed of carbohydrate, lipids, proteins, and certain amino acids. Or generally, we say that the cell wall is composed of peptidoglycan or murine. These are the chemicals which are present inside the prokaryotic cell, and as the name indicates, peptidoglycan, peptide for protein, glycan, gluco for glucose, mean carbohydrates. It is a conjugated molecule of proteins and carbohydrates which is present. It is also known as succulus. We will discuss the chemical composition of the prokaryotic cell in the lecture of the cell wall. And next, in the eukaryotic cell, the cell wall is present in protists, most fungi and plants, and is composed of chitin in most fungi and cellulose in algae and in plants, while it is absent in animal cells. Okay. Number three, respiratory enzymes are present on cell membrane. Respiratory enzymes are present on cell membrane because all membrane bounded organelles are absent. So mitochondria is also absent in prokaryotic cell, and if mitochondria membrane bounded organelle is absent, then we know that many bacteria exist which are aerobic bacteria. How will they respire? The question is that if a bacterium is aerobic but it lacks mitochondria, how aerobic respiration occurs? It occurs through the enzymes of respiration which are present on the cell membrane or plasma membrane invaginations, which are known as mesosomes. Inshallah, we will discuss in the next lecture mesosomes, etc. Next, these are absent on the cell membrane because proper mitochondria is present in the eukaryotic cells. And next, number four, thylakoids occur free in cytoplasm. Thylakoid mean actually the photosynthetic system and machinery. These are present in the cytoplasm region. These are also present in the form of membrane invaginations, but these lie in the cytoplasm region. But in case of eukaryotic cell, they occur within the chloroplast because chloroplast, a proper membrane bounded organelle, is present in eukaryotic cell. And next, number five is the cytoplasm lacks organelles like centrosomes. No doubt these are non-membrane bounded, but these are present. These are absent here. And next, endoplasmic reticulum, mitochondria, etc. All these are membrane bounded organelles. Membrane bounded organelles are absent in prokaryotic cell. While ribosomes are present, these are non-membrane bounded organelle. While in case of eukaryotic cell, all the cell organelles are present in the cell along with the ribosomes and other non-membrane bounded structures like centrioles, etc. So the eukaryotic cell will have a variety of different organelles due to the presence of all membrane bounded and non-membrane bounded organelles. And next, number six, gas vacuoles may occur while sap vacuole are absent. Sap vacuole here mean वो वाला बड़ा वैक्यूल जो प्लांट सेल में बड़ा सारा वैक्यूल होता है ना, वो वाला इन्होंने कहा यहाँ एब्सेंट है. हाँ, अलबत्ता गैस वैक्यूल्स हो सकते हैं. दीज हेल्प इन दी some sort of buoyancy or floating condition inside the water. While in case of eukaryotic cell, sap vacuole are commonly present, and this sap vacuole is basically a large vacuole which is present inside the plant cell only, and it is absent in animal cells. And next, number seven, 70 S ribosomes lie free in the cytoplasm or attached to messenger RNA. There are two important things. Number one is 70S, that the centrifugation properties, that the ribosomes are 70S, while in case of eukaryotic cell these are 80S particles. Okay, are you observing the last part? And next, the other thing is that that the endoplasmic reticulum, because it is a membrane bounded organelle, it is absent in the prokaryotic cell. So cytoplasm in this, so the ribosomes have to float inside the cytoplasm in free condition. These are absent on the endoplasmic reticulum because endoplasmic reticulum is not present. And number two, here the ribosomes are larger in size, and these are ATS particles, and these occur at 2 points or 2 locations. Number one, either floating in the cytoplasm, or these may be attached to the endoplasmic reticulum, and this endoplasmic reticulum is known as rough endoplasmic reticulum. Okay, so the two differences here described are: number one, the property of centrifugation, and number two is the location of ribosomes inside the prokaryotic cell, and prokaryotic and eukaryotic cell. Number 8, endocytosis and exocytosis do not occur through the plasma membrane in prokaryotic cell, and these processes occur in eukaryotic cells. Number 9, process of meiosis or gamete formation or true fertilization does not occur. Mean the cell division, the normal type of cell division, meiosis, mitosis, etc. These are absent. So the process of reproduction occurs without the process of meiosis and the proper fusion of the gametes. So you have to remember this point, that proper sexual reproduction is absent in prokaryotic cells. We use the term genetic recombination if some sort of bacterial genetic recombination occurs, bacterial exchange of DNA etc occurs, so it will be called as genetic recombination. We will not use proper term of sexual reproduction for bacteria, because meiosis does not occur, fusion of gamete etc does not occur. While in case of eukaryotic cells, proper meiosis occurs for the formation of spores in case of plants, and for the formation of gametes in case of animals. Gametes of plants and animals fuse together to form zygotes. So proper sexual reproduction occur in eukaryotic organisms. Okay. Next, cells are haploid. All bacterial cells are haploid. Remember this point: in their life cycle they are always haploid, and no diploid phase or some sort of zygote phase is observed here. And next, the cells are diploid, while haploid cells also occur. The somatic cells or general body cells are diploid, while their reproductive cells like spores or gametes etc are haploid. And next is the nuclear envelope. Nuclear envelope is absent, so that's the why we say that nucleus is absent. Basically, nuclear envelope is absent. It is an important concept. The absence of nucleus doesn't mean that they lack genetic material, DNA, RNA is absent in them. No, nothing. ये इस तरह ही है, आप इस तरह समझें कि जैसे जो है ना एक क्या आप कहें कि कोई आपके पास मिसाल के तौर पे ना कोई सब्ज़ी है, कोई चीज़ है, ठीक है। अब वो जब आप बाहर से लेके आ रहे हैं तो आप शॉपर में लेके आ रहे हैं, ठीक है। जब आप घर आ गए तो अब घर वालों ने उसको जो है ना किसी प्लेट थाली वगैरह के अंदर डाल दिया, तो जो भी आपकी सब्ज़ी है वो आपने शॉपर से निकाल ली ना। अब इस तरीके से आप ये तो नहीं कह सकते कि अब सब्ज़ी नहीं है, बस सिर्फ ये है कि शॉपर में नहीं है ना, वो किसी और जगह पे ऐसे ही जो है ना फ्रीली थाली के अंदर मतलब मौजूद है। बैंगन है, समझ लें, हैं थाली के बैंगन, हमारे यहाँ उर्दू में टर्म यूज़ कर लेते हैं। थाली के बैंगन है, वो थैली में थाली थाली के अंदर ऐसे घूमता फिर रहा है ना, तो मतलब बैंगन का वजूद तो ख़त्म नहीं हुआ। पहले वो शापर में था, वो फ्लोट कर रहा है, ऐसे मूव कर रहा है। अब ये यही कंडीशन जो है ना यहाँ पे कि डीएनए आरएनए वगैरह, मतलब जो जेनेटिक मटेरियल है वो, ये सब चीज़ें, पूरी मशीनरी ये यूकैरियोटिक सेल में न्यूक्लियर मेम्ब्रेन के अंदर बंद है, जबकि प्रोकैरियोटिक सेल में मौजूद है डीएनए, लेकिन वो न्यूक्लियर मेम्ब्रेन में बंद नहीं है, साइटोप्लाज्म में तैरता फिर रहा है। और इसकी प्रॉपर्टीज़ क्या हैं कि ये पहली बात क्या होगा, सिंगल होगा, एक ही होगा। दूसरी बात सर्कुलर होगा, यहाँ पे लिखा हुआ नहीं है, मैं ऐड कर देता, बल्कि उस वक्त जहन में नहीं आया। तो इसमें आपने याद रखना है कि बैक्टीरियल क्रोमोसोम इज़ नंबर वन सिंगल, number two it is circular molecule, while in case of eukaryotic cell the DNA molecule is a in the form of straight coiled strand which forms the chromosome in most of the cases. So the nuclear envelope is absent but DNA is present, and this area is known as nucleoid or chromatin body etc. All different names we will study inshallah in the next lecture. And here the nuclear envelope surrounds the nuclear material, proper nucleus is present, proper chromosomes are present. So next, point number 12, nucleolus is absent here. Nucleolus is a darkly stained body which is present inside the nucleus. And number two, one or more nucleoli are present within the nucleus. Nucleolus is a darkly stained body. It is absent in prokaryotic cell. It is present in eukaryotic cell. And number two, one or even greater number of nucleoli may be present inside the nucleus. And as you know that nucleolus is the factory of ribosome synthesis. And next, circular DNA is present without association with protein. This is an important concept. DNA is circular. Number two, histone proteins, which are present in the DNA of the eukaryotic cells to form chromatin material. These are absent in prokaryotic cells. Only DNA is present. While here the nuclear DNA is linear, not circular. It is linear. Number one, and associated with proteins which are known as histone proteins. And other than these, extra nuclear DNA is present without protein. What is extra nuclear DNA? Please tell me. Open your mic and tell me. Yes, please. What is extra nuclear DNA? जाग रहे हैं भाई सब लोग? Very simple. Extra nuclear DNA means DNA which is present outside the nucleus. Which organelles contain DNA other than nucleus? Please tell me. Are you here?
जी सर, अस्सलाम वालेकुम।
Walaikum salam. Yes, Javed. Please tell me.
जी सर, माइटोकॉन्ड्रिया और क्लोरोप्लास्ट।
Very good. Mitochondria and chloroplast. These are two organelles which contain DNA other than the nucleus. Okay. Remember this point. Next. Number 14, flagella, if present, are simple and consist of single fibril and formed of protein flagellin. And while here the flagella if present, these are complex. These have 9 plus 2 pattern of microtubules formed of protein tubulin. Tubulin protein जो है यहाँ पर है 9 plus 2 माइक्रोफाइब्रल्स की फॉर्म में. इधर जो है ना क्या है, यहाँ पे फ्लैजेलिन प्रोटीन है. 9 plus 2 अरेंजमेंट नहीं है, मेम्ब्रेनस कवरिंग में ऊपर से ये फ्लैजेलिन प्रोटीन बंद है. Next, plasmids and pili occur in many prokaryotic cells and these structures are absent. Plasmids are extra chromosomal circular rings of DNA. Other than main DNA, smaller rings of DNA are present in prokaryotic cells. These are known as plasmids. We will discuss these plasmids in detail. And next, pili are the projections which are present outside the cell wall. These are also present in prokaryotic cell only, and these are absent in eukaryotic cells. Next, most prokaryotes are asexual organisms because proper sexual reproduction is absent here, while most of the eukaryotes are sexual organisms. Okay, dear students. So this is a summary of the difference between prokaryotic and eukaryotic cell. And next, I'm sending you an assignment in the WhatsApp group, and you have to discuss back in the WhatsApp group this page. The contents of the page I'm sharing are very important, and please if you have any question regarding this presentation, you can